Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A Level Biology Unit 3 for June 2022. Let's begin with question 1. Question 1 says pollen grains will germinate and the pollen tubes will grow in water but not as quickly as they do on the stigma. This difference is thought to be due to several factors such as calcium ions, sucrose, and compounds containing boron. These factors are present on the stigma and in the style. The diagram shows a germinating pollen grain. We can see it here. There is the pollen tube, and we have the male nuclei. They are two, and here we have the pollen tube nucleus. So they say explain the role of the pollen tube in plant reproduction. In plant reproduction, you can see inside the pollen tube, we have the male nuclei, and we have the pollen tube nucleus. The pollen tube nucleus is going to cause the release of enzymes that are going to digest the tissues of the style in order for the pollen tube to be able to grow through. When this pollen tube is growing through, it's going to deliver the male gametes through the micropyle. I'll put an L here, micropyle. Let me put blue here, through the micropyle, and that is going to cause the fusion of the male gametes with the egg nucleus as well as the pollen nuclei, forming the final product of this process of reproduction. So let us continue. Here they say explain why pollen tubes grow in the same direction with the stigma after pollination. When they say here same direction, which direction and why, you have to put in that emphasis. So I said after pollination, the pollen tubes grow towards the micropyle. Remember, this is the direction. They asked you to explain, so we have to state which direction as well as why it is going in that direction. So after pollination, the pollen tube grows towards the micropyle. This occurs because the pollen tube nucleus controls the release of enzymes to digest the tissues of the style, and the embryo sac releases the chemicals. So that could be you earning the 2 points to get that. Next, they say the effect of sucrose solution of different concentrations on the growth of pollen grains was investigated. The percentage of pollen grains that germinated and the length of the pollen tubes were measured 1 hour after they were placed in the sucrose solution. They want you to state 2 dependent variables in this investigation. Remember here, they say the percentage of the pollen grains that germinated. So it means one should be the percentage of pollen grains that germinated, and the other should be the length of the pollen tubes. So these two, the length of the pollen tubes as well as the percentage of the pollen grains that germinated, are going to be the dependent variables. The dependent variables are the result of, from the change. When the change occurs, the change, different concentration, this is going to be the independent variable. The result when that change is made is going to be the dependent variable. So when we change the concentration of a sucrose solution, we're going to observe a change in the percentage of the pollen grains that germinate as well as the length of the pollen tubes. So these should be the dependent variables. Next, they say, explain why changes in temperature will affect this investigation. Any question asking you to explain why change in temperature, you have to think about enzymes involved in some of these processes. I say because the reactions leading to germination and growth are enzyme controlled. Enzymes like amylase increase the activity as the temperature increases due to higher kinetic energy. Guys, an increase in temperature leads to a higher kinetic energy, and therefore there is going to be more collisions between the enzyme and the substrate. I also went on to say, however, when the temperature is higher than optimum, denaturation begins to occur. Remember here they're asking you to explain why changes in temperature will affect. So temperature increasing will cause a higher rate. However, when the temperature goes beyond the optimum, then the enzymes are going to be denatured and there is going to be a decrease in the rate. So you have to consider both factors: higher temperature, higher rate of enzyme activity. However, when the temperature goes beyond the optimum, we'll see enzymes being denatured. Denaturation is when the 3D structure of the protein is changed as the bonds holding the 3D structure begin to break. So the active sites are going to be changed, and therefore the enzyme cannot longer, or the substrate can no longer fit into the shape of the enzyme, so the rate goes down. A solution containing calcium ions has to be added to the sucrose solutions. These solutions were made using a 2 mol per decimeter cube sucrose solution and a solution containing calcium ions and water. They want you to describe how you would make 10 centimeters cubed of 0.4 mol per decimeter cube sucrose solution using these 2 solutions. So the formula that is going to be suitable is this one here. I said you can say the concentration of the first solution times the volume of the first solution should equal to the concentration of the second solution times the volume of the second solution. This, keeping in mind, is the number of moles that are transferred from, in this case, the sucrose solution, are equal to the number of moles that are brought into the new solution during dilution. So, since this is, I took one to be the stock solution, which is the glucose solution. So, if this is the concentration of the first one and the volume, it means this concentration is 2 mole per decimeter cube, and this concentration is that volume is that. So, I made V1 the subject, which is going to be C2 times V2 over C1, and I substituted the respective values. Concentration 2 is 0.4 mole per decimeter cube. Concentration 1 is 2 here, I put it down here, and the volume is 10 centimeters cubed. I converted it to decimeters cubed because these concentrations are in mole per decimeter cube. So when I use my calculator, I got 2 times 10 power negative 3. This is decimeters cubed. I converted that to centimeters and it gave me this. So it means in order to make 10 centimeters cubed of a specific solution, I have to add 2 centimeters cubed. 10 centimeters cubed of 0.4 mole per decimeter cubed, you have to add 2 centimeters cubed to 8 centimeters cubed of the calcium ion water solution. So here I say, so to obtain 10 centimeters cubed of 0.4 mole per decimeter cubed sucrose solution, add 2 centimeters of sucrose solution. This is the original stock solution to 8 centimeters of calcium ion and water. Then you'll be able to obtain the specific concentration you're looking for. Next here they say the calcium ion solution is alkaline. State how the pH of the sucrose solutions used in this investigation could be adjusted to a required value. So here I said by diluting the solution. Remember, this is pH. You could dilute the solution, or you could use a specific buffer that can ensure that the pH is maintained at a specific value. So let's continue. Here they say the graph shows the results of this investigation. We can see here they have the percentage germination, and here we have the sucrose solution, its concentration in mole per decimeter cube. We can see the percentage germination increases, and then say this is rapid decrease, rapidly decreases. For this one here, it decreases and then decreases rapidly, and until we can see there is 0% growth. So they asked us, they say, draw a suitable table to show the results for percentage germination. So I just went into the graph. You can see at 0 here, remember this is percentage germination. So for sucrose solution, this one here, I'm using this one here. At a 0, we can see here we have 0, it's about 6. And then when we go to 0.2, we have some value which is 46. And so on. So we were taking these values here, those values until here. Those are the values I put in the table. And you can see at 1.6, we have 0% germination. So you remember in a table like this, you have to put the labels as well as the units and put the values that are correct, and everything will be okay. Moving on. Here they say comment on the conclusion that both percentage germination and mean pollen tube growth have the same optimum sucrose concentration. I say, according to the graph, both have the same optimum concentration of 0.4 mole per decimeter cube. However, the actual optimum concentration for each could be above 0.2 mole per decimeter cube, but below 0.8 mole per decimeter cube. Both could have the same optima, meaning same optimum. So the experiments could be repeated with sucrose concentrations between 0.2 to 0.8 mole per decimeter cubed in order to confirm the optima for each. Before I finish this, I want to take you back. Remember, we have this gap here. There is this value here. We know the exact value at this point. We also know the exact value at this point. However, we are not sure if at this point the value should have been here, but because we didn't carry out this experiment, we didn't find the value. It could be even here at this point, but we didn't do that experiment. So we need to do more experiments within this specific range here in order to know if they have the maximas at the same point or their maximas are varied, and how high are the maximas actually. So repeat the experiment in this region and then be able to confirm if it's true or if it's not. This brings us to the end of question 1. Let us continue to question 2. Question 2, Psychotria species are rainforest plants that are widely used as herbal medicines. It has been suggested that extracts from these plants could be used to inhibit cell division. The effect of extracts from 2 species of this plant on the mitotic index of onion roots was investigated. 2 concentrations of extract is we have 1 milligram per centimeter cubed and 7 milligram per centimeter cubed were made from each of the 2 species of Psychotria. We have species A and species B. Now, extracts from one of each of these was used in the experiment. So they say onion roots were grown in each concentration of extract and a control as shown in the diagram. We see this is the onion. Those are the roots that are grown in the extract solution. So the setup was like this, and after 24 hours, the mitotic index was determined for these roots. Let us continue. Here they say describe a safe method to observe the stages of mitosis in these roots. Because they have said it has to be a safe method, you have to wear safety gloves as well as goggles. And then we have to cut the root tips. Remember, this is an experiment about mitotic index, so the root tips have to be cut, and these root tips have to be placed into warm acid. Then after that, the root tips have to be placed in a staining agent like acetic or stain, or you could use methylene blue. Then place those same root tips on the microscope slide and macerate, or you could squash. Using a high power microscope, you could observe the tissues for cells in mitosis. So in this case, you can observe to see if the cells are going through mitosis or not. The next part, they say the photograph shows part of a root tip from this investigation. They say calculate the mitotic index of this root tip and give your answer to 2 significant figures. Now, I want to enlarge this a little bit so that you can see. When we observe, we can see this is in mitosis because we can see the chromosomes properly. This is and that is. This 1, 2, 3 can easily be observed, but we also have this one that could look as if it's still in mitosis. So a student could use 3 out of the total that is. I counted the total and observed there were 40, or they could use 4 out of 40. So for me, I used 4. Mitotic index should be the cells going through mitosis divided by the total number of cells times 100. So 4 divided by 40 times 100 gives me 10%. Remember they say 10.0. Remember they say 2 significant figures, so it has to be 10.0%. That is what I have, and my answer is that. So moving on, here they say the table shows data from this investigation, so we can have for each different species, we have species A, that is the information, species B, that is the information, and the control, that is the information. So they want us to plot a suitable graph to show the data in the table. So remember, this is mitotic index. On the results are mitotic index, so I put it on the vertical axis, and the horizontal axis, I put the specific concentrations of each species. So for species A, we can see here, 1 milligram per centimeter cubed and 7 milligram per centimeter cubed. And for species B, we have 1 milligram per decimeter cubed and 7 milligrams per decimeter cubed, as well as the control that had 0 milligrams per decimeter cubed. Now, they wanted us to plot a graph, so I plotted it as a bar graph because we can see here it was 4.7, and then down here we can see it's 1.2. This value here, it's 4.2, then this is a 1.9, and finally we had 7.4. So if you represented your values as bar graphs, then it's going to be easy for you to observe this. So I hope this can be visualized here. We can see the plot was of a suitable graph to show the data in the table. I drew the vertical axis is the mitotic index, and the horizontal axis is the extract concentration per source. Remember, we had source A as well as source B with their respective concentrations, and the bar graphs are representative of that. When you draw a bar graph, you have to leave some space in between in order to cater so that they do not look so connected or contacted together. Let's continue. So here they say describe how this investigation could be modified to determine if the differences between the mitotic indices are significant. Any experiment that is carried out, if you want to establish significance, you have to repeat the experiment, calculate the mean from those, and then you have to keep the conditions constant while you repeat those experiments. When we do this, we have to use the calculated mean as well as the highest and the lowest values for that specific condition in order to add error bars to the values or the bar graph we have drawn. Then we will check for presence of overlap between the error bars of the different values. If the error bars, let's say, of concentration A or for one species and the other species do overlap, then we could say the results are not significant because the error bars do overlap. However, if there is no overlap, then we can say these error bars do not overlap, so the results should be significant. Significance is established if you repeat the experiment, calculate the mean, put error bars, and see if there is overlap or not. So this brings us to the end of question 2. Let's go to question 3. Question 3, many food crops such as potatoes and bananas contain starch. The starch content can affect the taste and cooking properties of food. Potatoes can be boiled or baked. Potatoes with high starch content are good for baking, and those with lower starch content are good for boiling. It is suggested that King Edward and Rosette potatoes are better for baking, and Nicola and Purple Congo potatoes are better for boiling. A student decided to test these suggestions. So it's like a hypothesis that you're trying to test around. A standard set of starch solutions was prepared and tested, and the table shows the results. So we have, if we have a concentration of 1.0%, it's going to be black, 0.1%, it's going to be dark blue, 0.01%, it's going to be pale blue, 0.001% very pale blue, and 0.0001% is going to be pale brown. This is telling you that if something is black, it has a higher concentration of starch, and if something is pale brown, it has a lower concentration of starch. Think about this as if we are establishing a standard. So maybe we're going to use these colors to compare if the concentration is high or lower by observation. So let us go to the next page. Here they say, describe how the data in the table could have been obtained using 1% starch solution. I want to take you back to the table. The table has different percentages or different concentrations. So if we began with a 1%, we are going to carry out serial dilution. So this is dilution. So by 10 is 10, 10, 10 until the end. So here I say, you carry out serial dilution using the 1% available stock solution. So this part here is explaining the details of serial dilution. I don't know if you guys can see here, I try to place it here. So if you have the 1% stock solution, let's assume this, you take a 1 ml from here and add it to 9 ml and then mix it thoroughly to create a new concentration, which is going to be a 0.1%. Then from the 0.1%, take 1 ml and add it to 9 ml. This 9 ml has to be distilled water to make a dilution, which is going to be 0.01% after you mix. Then from here, take out 1 ml, add it to 9 ml of distilled water, and then mix in order to create that. And to the final one, carry 1 ml, add to 9 ml, mix thoroughly to create that. This is what we call serial dilution, when you pick from the previous concentration and create a newer concentration, which is more dilute in comparison. So here I say, this is the detail. To 1 ml of 1% solution, add 9 ml of distilled water to obtain the 0.1% solution. Then transfer 1 ml of the 0.1% solution to 9 ml of distilled water to obtain the 0.01% solution. Continue this trend 2 more times to obtain the 0.001%. Then I went on to say, to each test tube of starch solution, add iodine of the same concentration and volume. Remember, if the iodine concentration is not the same, then results could be different. So add the same volume of iodine, and that iodine should have the same concentration, and then observe the color of the resultant solution in the test tube, similar in diameter. The test tube has to be similar in diameter so that the results are not varied, or so that there is no confusion as you observe. So I said, note down the observed color of the solution, and that will be it. So we carry out serial dilution, create the various concentrations, you add iodine, and then you observe the colors that are going to come out of that. Let's continue. So here they say the table shows the results of the 4 potato varieties. So we have, they have the King Edward. So from the real experiment, the King Edward was black, meaning it has a higher concentration of starch based on what we saw. Back here, these colors here they're using for comparison. So if it's black, it means it has a higher concentration of starch. Let me go back. So this is black. Then we see the Nicola, which is very pale blue. Then this is pale blue, and this is dark blue. We can see black and dark blue should have a higher concentration of starch, and the pale blue and very pale blue, the starch content should be lower. Remember again from the question, they said if there is a high starch content, they are going to be good for baking, and if they have low starch content, they're going to be good for boiling. So I can observe that this one here and this one here, since they have a high starch content, they are going to be good for baking. However, the King Edward is going to be the best for baking because it has the highest concentration of starch. These ones here are going to be good for boiling. However, this one is going to be the best for boiling because it has the lowest concentration of starch. So let us see. They say discuss the extent to which these results support the suggestion about the use of these potato varieties. So I say, based on the table of results, King Edward and Rosette potatoes show higher starch content because one is black and the other is dark blue. They should therefore be used for baking. Remember, we are referring to the data from the question. Then also, the Purple Congo and Nicola show lower starch content because they are pale blue and very pale blue, respectively, so they should be used for boiling. Also, based on the data, King Edward has the highest starch content, so it's more suitable for baking than Rosette. So I say, since the results are based on a color observation, this is a semi-quantitative method because you're observing, and actually it's going to be subjective because different people have different visual capability, and that could cause errors or misunderstanding of the results. So you have to be very careful not to make erroneous conclusions from your experiment. So let's move on. Here they say suggest how this investigation could be adapted to produce quantitative data. Quantitative data, it means more accurate or more dependable data. So because this is based on color, you could use a colorimeter to measure the absorbance. Remember, if these are colored solutions, they're going to have different absorbances depending on the color present in the solution. So the cuvette has to be the same, and then you have to use a colorimeter to be able to establish if the results are actually accurate as you had observed. Next here they say it is suggested that the change in the taste and consistency of stored bananas is due to changes in their starch content. An investigation of the content of starch in stored bananas produced the following results. So we can see as the storage time increases, of course the starch content is going to decrease, but the decrease is not uniform. At first we see a slow decrease, then we can see it's a little bit slow, then it a little bit increases, and then slow, and then it goes rapid. So it changes over time based on, of course, the time in storage. So here they say calculate the rate of decrease in starch content from day 15 to day 23. This is day 15 and this is day 23. So I had to get the starch content at day 15. You can see that is 14 AU. And then at day 23, we can see we have 5 AU. So the difference in the starch content is 14 minus 5, which gives us 9 AU. Also, I need to find the difference in the number of days. It's day 15 and day 23. The difference in time should be 23 minus 15, which gives us 8 days. So here we can see the rate is equal to the decrease or change in starch content divided by the time, which gives me 1.125. So I give my answer in that, 1.125 AU per day. That is the answer. So we continue here. They say describe the changes in starch content during the 23 days of the investigation. This is to describe, so you have to talk about how the trend changes. Overall, I said there is a non-linear decrease in starch content with time. Let me take you back. Of course, we can see it's non-linear overall. There are changes that occur. So from 0 to 5 days, the decrease was little. The decrease further slowed down between 5 to 15 days. And after 15 days, there was a rapid decrease until day 23. That is what we observed. And since this is a 2-mark question, any 2 of these would have earned you the marks you're looking for. So lastly here they say, sketch a graph to show the soluble sugar content of the bananas during storage. So this is going to be based on the graph you have this side. Of course, we can see there is a decrease. This is going to be less. So we expect that sugar that is in, or the sugar that is in the solution to be a little bit less. Then there is a little bit slower decrease here, and then we can see the rapid decrease. So from day 15 to day 23, the graph you have to draw should be higher. Like you can see here, day 15 to 23, I drew a little bit higher. You can see there is a bigger increase. So initially we have less. Then it's no longer steep, it's almost constant. The reason for this is, yes, it's less, but because it's overall sugar that has linked overall, it's going to be a little bit higher than the initial. because yes, even if it's going to be a lower increase, there is still some that is being added to the solution. So afterwards, after day 15, there is a very big increase in the sugar content that has linked into the solution. So that is why I drew it like that. The graph has to make sense the way you draw it. It doesn't matter if you are drawing it in a specific way, but the trend had to make sense. There had to be a very high, a steep change from day 15 to day 23. So this brings us to the end of this paper. Thank you for being with us. Please do not forget to subscribe. See you in the next video. Bye-bye.